Welcome back to our explainer on the FIM method. If you caught part one, you know we left our plant in, well, a state of total botanical chaos. Its main command center was just partially destroyed. That apical dominance we talked about, shattered, but not entirely removed. The plant is literally left in a state of sudden, absolute confusion. So today, we're jumping right back in to unpack the microscopic biology of exactly how the plant responds to this structural crisis. And we're going to look at the underlying systems that make this technique so wildly fascinating. So we've gone ahead and partially damaged the shoot apical meristem. Now what? From the plant's perspective, the command center hasn't actually completely disappeared, like it does when you cleanly top a plant. Instead, it's just heavily damaged. Some of those crucial cells survived the cut, while others definitely didn't. The plant now faces a highly unusual challenge. It has to make a choice. Can this damaged meristem keep functioning, or is it time to let totally new growing points take over the operation? Unlike standard topping, where the path forward is pretty clear-cut, FIM creates an environment of total biological uncertainty. To really understand this choice, we have to zoom in and look inside that growing tip at something called the shoot apical meristem, because ultimately, it all comes down to stem cells. Now, when we hear stem cells, we almost always think about human medical research or animal biology, right? But plants have them too. The shoot apical meristem is essentially this tiny, densely packed reservoir of completely flexible, uncommitted stem cells, and they continuously divide throughout the entire life of the plant. Every single new leaf, every node, every flower, and every branch you see, it all begins as one of these microscopic cells. Because they haven't committed to becoming one specific type of tissue yet, they are totally flexible. Given the right hormonal signals, they can become almost anything the plant needs to survive. To put that into perspective, think about animal stem cells. They generally have pretty limited, highly specific regenerative abilities. But plant stem cells? Plants have been using these to completely reorganize their canopies for over 400 million years. This evolutionary trick gives plants an absolutely incredible advantage. I mean, if a plant loses a branch, it just grows another one. If it loses leaves, it pushes out new ones. If its growing tip is damaged, like what happens during our FIM cut, it possesses the stunning ability to completely reorganize its entire canopy structure. It's literally a major reason they've survived constant grazing from animals, heavy storms, and sweeping diseases for hundreds of millions of years. Imagine taking the CEO of a major company, the plant's single dominant shoot, and abruptly replacing them with several mid-level department managers who are all trying to lead the company at the exact same time. That's what's happening here. When you FIM, instead of removing every single stem cell in that apical meristem, you end up leaving behind several isolated groups of survivors. Each of those surviving groups still possesses the inherent ability to grow, but without the CEO in charge to keep them in line, none of them completely dominates the others. So instead of producing one dominant shoot, the plant starts pushing out several competing shoots simultaneously. It's basically total management chaos. But you know, stem cells are really only half the story. Hormones are the ones actually pulling the strings. It's this epic, invisible, botanical tug-of-war. On one side, you have auxins, pushing a strong top-down signal to suppress lower buds. On the other side, cytokinins are rushing up from the roots to wake those buds up and encourage cell division. When a FIM cut happens, that strong, highly organized auxin production gets completely disrupted. Suddenly, you have several weaker auxin signals just competing with one another. Meanwhile, those cytokinins, they just keep rushing up from the roots unchecked. Without that one overwhelming auxin source keeping things locked down, cytokinins gain the upper hand, and growth points all over the plant start waking up. And that brings us to these completely hidden botanical gems, axillary meristems. These are tiny, often unnoticed structures sitting quietly in the little angle between each leaf and the main stem. They are already there, just waiting for their moment. Most of them remain totally inactive, while that main apical shoot, the CEO, dominates the plant. But once that FIM cut weakens the apical dominance and those cytokinins flood in, these hidden meristems wake right up and accelerate their own development. This is fascinating because some of the explosive extra shoots that growers attribute directly to the damaged tip might actually be originating from these previously suppressed axillary meristems springing into action. Now, because there are so many hormonal and structural variables at play, we really need to address a major beginner misconception right here. FIM guarantees 4 new tops. Actually, scratch that. It definitely does not guarantee 4 tops. Yes, 4 tops are entirely possible, but the variety of outcomes is huge. 3 tops are extremely common. Sometimes you only get 2. Occasionally, just 1 growing tip survives if the cut was too low, which means you essentially just topped the plant by accident. And if the cut is too high, the original meristem might just shrug it off entirely and continue growing almost normally. Because the plant is desperately attempting to repair itself and redistribute growth at the exact same time, the outcomes are incredibly varied. So, what's the core takeaway here? Biology rarely produces identical outcomes from identical treatments. A plant is not a factory assembly line. Everything interacts simultaneously. Different cuts preserve different portions of that apical meristem. Different cultivars possess entirely different branching tendencies naturally, and different environmental conditions heavily influence that delicate hormone balance we just talked about. Every single plant is slightly different. Every microscopic meristem is slightly different, and every snip of your scissors is slightly different. From a strictly scientific standpoint, FIM is inherently wonderfully unpredictable. And honestly, this unpredictability completely divides the cultivation community. If you're managing a massive commercial facility with thousands of plants, you absolutely need consistency to simplify your labor scheduling and canopy management, which is exactly why commercial growers almost always prefer the totally predictable results of standard topping. But for home growers, discovering how each individual cultivar responds is part of the fun. Embracing the unpredictability of FIM makes it a fascinating hands-on experiment. One plant might surprise you with 4 vigorous shoots, while another teaches you something completely different about its own unique genetic tendencies. Which naturally leads us to the question that always makes experienced growers crack a smile. Exactly where should I cut? It is, without a doubt, the most common question beginners ask when they're first trying to replicate the FIM technique. They want a specific measurement, a perfect angle, a guaranteed formula to get those legendary 4 tops every single time. Basically, they're looking for an exact structural blueprint for a technique that was, by its very nature, born completely from a mistake. The honest answer to that question? There just isn't one perfect location. Remember, you are working with incredibly complex living tissue measured in mere millimeters. Because biology flat out refuses to follow simple rules, even the tiniest micro differences in where your blade lands will produce entirely different biological responses. You might hit a cluster of stem cells perfectly on one plant and miss them by a fraction of a millimeter on the very next one. That microscopic variance is exactly what makes the FIM method both incredibly frustrating to master and completely fascinating to execute. By now, you might be noticing a broader theme connecting all of this plant training together. Whether we're talking about low-stress training, supercropping, topping, or FIM, they literally all manipulate the exact same foundational systems: hormones, light, resource allocation, and that incredible flexibility of plant development. The physical techniques might look completely different to the naked eye, sure, but the underlying biology connecting them is remarkably similar. You are always playing with that delicate balance of auxins and cytokinins and telling the plant exactly where to send its resources. Okay, we've unpacked the biology, we've explored the stem cells, and we've mapped out the hormones. But I want to leave you with the ultimate question. Is FIM actually better than topping? Well, in part 3 of this explainer, we are going to settle this debate once and for all. We'll compare the hard data on yield potential, look at recovery times, and explain exactly why experienced cultivators eventually swear by one technique over the other based on the specific genetics they're growing. So think about what we learned today, get ready to apply it, and I will see you in the next one.